Okay, so we talked about cell cycle quite a lot already, but the issue is that quite often you might have a more homogeneous cell population, or within one cluster of one cell type, you might see a clear separation by cell cycle like this. And by removing the cell cycle effect, you can instead find them to separate by cell type. So one part is to try to predict cell cycle. So we already did this in the QC lab. In Seurat, we have the cell cycle scoring function that takes a list of human cell cycle markers for G2M and S phase, and basically just calculates how highly expressed a set of genes is versus a random set of genes per cell. And then it does the same for the S phase gene. While Cyclone uses relative expression of pairs of genes. So it basically takes, it's trained on cells that are sorted by cell cycle, looks at this gene should always be higher than that gene if it's S phase, for instance, and then uses those pairs instead. In ScamPy, they use the same implementation as in Seurat. So we get these scores. And then it's still just a prediction, of course. So you have to at least look at the scores, plot them, and see that they make sense in your data set. If it looks like everything is predicted to be S phase and that doesn't make sense given the data, you know, that maybe it's not a good idea to take them and regress it out. But to remove the cell cycle effect, linear regression works quite fine. There are some more advanced, like SciLVM, they use these latent variable models, and also CC remover, OSCOPE, ReCat. I think for me, these give fairly similar results still, and it's not always the case that you manage to remove the cell cycle effect even though you do this. That could be an effect of the scores not being calculated correctly, or that there's additional signals in the data that's so strong that you can't remove the cell cycle effect. I found, especially working with like cancer data, it's almost impossible to remove the cell cycle effect in a cancer data set completely.